This is Lecture 7, Microbial Genetics, and what we'll talk about in this lecture are various aspects of the genetics of microorganisms that, for the most part, affect our understanding of them and affect how we relate to them. So I'll start with some kind of basic genetics, and then you'll see a lot of examples that kind of explain some of their behavior and why they are problematic to handle in clinics. So step by step, we will get there. So where I want to start is with gene regulation, and that's what's shown on this first slide. But I want to pause for a minute and turn off the video and just ask you, what is gene regulation? What is gene expression? Think back to any cell. Any cell has a job to do. Any cell in a human body has a job to do. And it does that in part by having specific proteins that give it specific abilities. But cells don't do the same thing constantly forever. Sometimes they're active, sometimes they're quiet, sometimes they grow. Sometimes they repair proteins, other times they make proteins. Well, how do they control all of this? Usually, they control it by controlling which proteins they have and which proteins they make. And so when I talk about gene expression or gene regulation, what I'm talking about is how any type of cell controls which proteins it makes. So what we can do is compare what you know about human cells, like this one over here on the right, on the left, to bacterial cells. And this is laying out for you the, central dogma of biology, where we have something like DNA that is transcribed to make RNA, and that is translated to make a protein. And all those steps are steps where it would be hypothetically possible to change the rate at which a protein gets made. Because you can imagine if we slow down the process of transcription of a certain gene, we're going to slow down all of this. If we slow down how fast it's translated, again, we slow it down. And so anytime there's a step that we can look at, like transcription, processing, translation, post-translation, all of those are opportunities to control how fast we make each individual protein. And so what they've done in this diagram is drawn these arrows, and the thickness of the arrow is supposed to correspond to how much regulation happens on that step. So looking here, what we see is that the thickest arrow is transcription. So our cells would decide to transcribe or not transcribe a gene, and that's a big part of how they control whether a protein gets made or not. But also, they control the rate of RNA processing, of like splicing and capping and polyadenylation and things like that. And at all of these steps, these genes can be regulated. Well, we compare that to a bacterial cell where it's a very different picture. The first difference is that there's no pre-mRNA. There's not necessarily splicing or capping or polyadenylation in bacteria. Bacteria have a kind of simpler genetic system. It still uses DNA, RNA, and proteins, but the messenger RNAs don't need to last a long time like they do in eukaryotic cells. So there isn't an RNA processing step. And the other thing I'd point out is that almost all the regulation happens at the level of transcription. So in bacteria, if you understand how they control whether or not they transcribe a gene, you are understanding how they control how much of a protein they make. Because very little happens downstream of that. Most of the regulation happens at transcription. And so they can control like a yes or no, does the gene get transcribed or not? But more often what they're doing is controlling how often it gets transcribed. Is it transcribed once an hour, once a day, once a minute, that's what they control. And so in bacteria, transcriptional regulation is the main part of the regulation. Keep that in mind, and we'll look at kind of an interesting genetic feature from bacteria called operons. So what I'm showing on this slide, this is supposed to be DNA in a bacterial cell. So DNA is a long strand, a very long strand. And if you zoom out and look at it when it's kind of supercoiled, it's basically a long rope-like structure. But we could look at DNA, and all along its length there are going to be genes we could see. So if we were reading the sequence of the DNA, we would be able to identify areas where there are genes. There are other areas also, certainly areas with no genes. There are areas with regulatory functions. So like a promoter is where it's the DNA sequence where various regulatory proteins bind to initiate transcription. And so this sequence of this promoter could be a strong promoter that causes transcription to happen very quickly, or it could be a weak promoter. That's what we would call a promoter that causes transcription to be rare. So there's that. And then what bacteria do, well, what any cell would do is downstream of this, that's where the gene would be. So the transcription start site would be right here. An RNA polymerase would bind here, and it would move this way and transcribe the gene. And so that happens in bacteria. But what bacteria do is often group multiple genes together, and they share a promoter, and they share a transcription start site. And what happens is RNA polymerase binds here and goes this way and transcribes, but it transcribes the whole thing as one big messenger RNA. And this is very common in bacteria. In some cases, there will be 2 genes in an operon. In some cases, there will be 5. But many times they transcribe multiple genes using one transcriptional regulator and one transcriptional initiation site. So I'll show you an example of this and show you kind of why they do this. The thing to think about is that bacteria have to rapidly react to a changing environment. So there's a well-known example of this called the Lac Operon. And a lot of times in introductory biology classes, they will teach you the Lac Operon because that is an example of genetic regulation that's simpler than a lot of what they could show you in eukaryotic biology. I'm not that interested in the details of this. What I'm interested in is showing you the purpose behind it. So E. coli, this is an E. coli operon. E. coli can use lots of different sugars for carbon, energy, and electrons. But E. coli prefers glucose. So if glucose is present, E. coli will use it. If glucose is not present in the environment, it will use whatever other sugar it can find. It needs specific enzymes to break down sugars. It needs the glycolysis enzymes to break down glucose. It needs other enzymes to convert things like lactose into glucose so that it can use glycolysis. So there are very specific proteins the cell has to make if it wants to use a different sugar, like lactose. So these are the genes E. coli would express if E. coli wanted to use lactose as a carbon source. So if it detects lactose in its environment, it will start transcribing these genes. And the reason this is in an operon is because E. coli wants to very quickly make all 3 of these as soon as it detects lactose. So it's going to start transcribing all of them. And what you'll see once transcription happens is the messenger RNA molecule is going to have all 3 genes. But then what you'll see is ribosomes will attach to this messenger RNA. So remember, RNA polymerase attached here, and it transcribed the whole operon. Well, you're going to have separate ribosomes for each gene. So translation is going to happen separately for each of these. You're going to make 3 separate proteins. And the proteins are different sizes, and the cell can choose to have more of this one than it makes of this one, or vice versa, because each one is independently regulated in terms of translation. So none of these are made if transcription doesn't happen, but once that happens, the cell can choose how much of each protein it wants. And so translation will lead to all these different proteins and enzymes. One detail I do want you to know from this slide is how the names are written. So let's look at LAC-Z. This is a typical name for a bacterial gene. There's a 3-letter abbreviation that says something about what the protein does, and then a random letter attached to it. So genes from the Lac Operon are LAC-Z, LAC-Y, and LAC-A. They're out of order because they were discovered out of order. Biochemists discovered these enzymes long before they could sequence DNA. So that's what happens. Okay, so take a minute to go back through this and understand it. I would highly recommend that. But then from here on out, we're going to be looking at proteins in DNA and mutations. I want you to keep in mind that a cell is constantly trying to decide what protein it needs to make, and to make that protein at the appropriate concentration and rate. So we get into it. Think back to protein structure. What is it that makes 2 proteins different from each other? What is it that gives them their functional abilities that are specific to actin or myosin? What makes actin and myosin different? Well, the first thing is their primary structure, their amino acid sequence. This is what this is what the DNA encodes. It's the amino acid sequence. What we find is as soon as the protein is synthesized, it will immediately start folding into small structures like sheets and helices that might be 10 amino acids long. Well, there might be 1,000 amino acids in the protein. So these would make up tiny little regions within the protein, but they start to give it 3-dimensional structure. And then, once those are made, they interact with each other to make the complex 3-dimensional folded shape that any protein needs. And that gives it the perfect shape of its catalytic sites, if it's an enzyme, or the perfect shape for it to bind something if its job is to attach to something. So that's called tertiary structure, is that folding, that 3-dimensional shape. And then quaternary structure would be for any enzyme that is more than one subunit stuck together, so more than one separate protein molecule that are attached and work together. That's called quaternary structure. So we won't talk about these things again, but again, I want to make absolutely certain that you understand that the primary structure of a protein, the amino acid sequence, encoded by the DNA sequence, that is what determines a protein's structure and function. And that's important because we're going to look at mutations. And when we look at mutations, we have to think about what is the effect of a change in the DNA? What happens to the cell if the DNA changes? And in almost every case, we have to look at what happens to the proteins encoded by that DNA. So let's think about this a little more. Let's think about how do we connect DNA to proteins? How does DNA encode the amino acid sequence? Well, you might remember seeing a codon table like this. it shows for any 3-base codon you might see in DNA or the messenger RNA, which amino acid it encodes. So for example, if you find, an AUG, that's usually the start of a gene. And then if what you find next is ACA, find ACA. So ACA is going to be in this big row, and then C up here, it's going to be in this column, so it's going to be here somewhere. And then over here, we find A. So ACA, well, that encodes threonine. And you certainly don't have to memorize this, but you should know what is telling you. You should know what kind of information you get from this. And this is how we would figure out from a DNA sequence what the amino acid sequence is. We would use this exact table. You'll see lots of different versions of it that are formatted differently, but this is the basic idea. So this brings us to mutations. And the idea with a mutation is that one base might change. So it might go from ACA to AAA. If that happens, this threonine is no longer part of the gene. Instead, this AAA encodes a lysine. And that has different chemical properties to threonine. That's going to be a different molecule, and it's going to interact with the world in a different way. And so if you change the ACA in DNA to AAA, well, that is going to change the protein. And that's what happens with mutations. So this is, again, this is things I've said so far, sort of the basics that I want you to think about that connect DNA to proteins. And anytime a DNA sequence changes, that's a mutation. Often they happen when DNA is copied and there's some kind of error. And so a lot of mutations will be one base, like what I just showed you, where a cytosine changes to an adenine. That certainly can happen. But in other cases, much bigger changes can happen. So part of a gene could be deleted, or it could be doubled. There are a lot of cases where a big piece of DNA accidentally gets doubled, so there are 2 of them in a row. And these things can all have huge effects on cells by changing which proteins get made. So mutations, if they happen in an essential gene like actin, if there's a mutation in the gene for actin, an animal will not survive past fertilization. If there's a mutation in a less important protein, it might be possible to survive with that, and a person might not know it. So the most severe kind of mutation is going to kill the daughter cells. So when the daughter cells are made, they can't survive. That's severe. But there are other mutations that don't change the amino acid sequence at all. So we'll see some examples of that. So we can kind of lay out the 5 types of mutations and look at them. So make sure you learn what these are, these types of mutations. These first 3 refer to situations where one nucleotide changes in the DNA, just one nucleotide changes. And so I'll start down here with a silent mutation, and that's when the DNA changes, but it doesn't change the amino acid sequence. Well, if that happened to one of your cells and then it divided, you wouldn't know it. It's a silent mutation. It doesn't affect anything on any short-term basis. The sort of mutation I showed you a few slides back was a missense mutation, where the amino acid changes because of the mutation. And there's a really severe type of mutation where a stop signal happens, where the mutation changes from an amino acid to a stop signal. That means when it's time to translate that gene, the ribosome will stop wherever it hits the stop codon, and it won't finish making that protein. So the protein could be completely inactive. And then there are situations where DNA gets inserted or deleted. And this can matter because the reading frame can change. So let's look at all of these. We're going to start with this normal sequence up here. So it starts with AUG, that's the symbol for starting a protein in bacteria, and then glutamine, threonine, and serine, and then a stop codon at the end. So we're saying that this, I guess, 4 amino acid peptide is what the cell wants to make. Well, the silent mutation would change this G to an A. And if you go back and look at the, If you go back and look at the codon table, CAA and CAG both encode glutamine. They both encode glutamine. So it wouldn't change anything to change this to an A. But if we changed this to an A down here, now instead of CAG, now it's AAG, and that's lysine. So that is going to definitely change the peptide. Glutamine and lysine are not the same, and they will have different chemical properties. So this missense mutation will change what this peptide does. And then here's the severe one, the nonsense mutation. Instead of CAG glutamine, it's UAG, which is a stop. And that means none of these other amino acids even get incorporated into the protein. It just stops at this point. And insertions and deletions can be equally severe because they change the reading frame. So if you look here, what's happened is one base has been deleted. So this A has been deleted. So what happens when the A is deleted, basically all of these move over 1, but the cell keeps reading this 3, then this 3, then this 3, then this 3. So we don't see a problem here. That hasn't changed. This hasn't changed. But then when we get to here, instead of ACA, it's CAU, because these 3 have moved over 1. And CAU encodes histidine, and that is not the same as the threonine that was supposed to be there. And all the downstream proteins are basically scrambled and randomized when this happens. So an insertion or deletion will usually completely randomize the gene because it changes the reading frame. So again, what I would say is, if I lost you, pause this, go back, make sure you understand it, because this is important. You really should understand the connection between DNA mutations and what happens to the protein, because we're going to come back to this and use this information to think about evolution and the rapid, scary evolution of microorganisms. So I will see you in the next video.